Okay, I'd like to have everyone's attention. We're on to Industry Updates 2, Part 2. So I gave you the introduction, so I'm sure you all have it memorized. And now we're going to hear from 5 dynamic representatives from the companies, and I'm going to let them take it away. Come on up.
Hi there, good afternoon. My name is Ash Dugar. I'm with Dyne Therapeutics. I'm the Chief Medical Affairs Officer. First, I just want to say it's a privilege to be talking to the community living with myotonic dystrophy. It's always a privilege to talk to you. It's incredibly insightful, very humbling, and really I want to thank Tanya and the organizers at MDF for inviting us to speak here today. I have a bunch of slides. I'm going to go through them fairly quickly because Tanya did not give me 30 minutes to speak, right? Okay. These are our forward-looking statements. You can refer to our website if you're curious. I always like to begin with this slide. At Dyne, it all starts with our mission, which is to develop life-transforming therapies for people living with serious muscle diseases, with myotonic dystrophy type 1 being a priority. This is truly core to who we are and what we do each day. I'm not going to go through the disease. You've heard amazing talks from Drs. Berglund, Johnson, other speakers over the last couple of days. And for me personally, listening to the mini PFDD with the patient panel and others was really enlightening and very moving. And it was, again, providing insights for us that we just can't get reading, papers and even talking to as many physicians as possible. So we really want to thank you all for sharing your stories. Again, others have gone through the genetic basis of myotonic dystrophy type 1, so I won't go through it here. What I will say is that the term that keeps coming up over and over in terms of the disease is this term spliceopathy or splicing defects. It's this missplicing that really leads to the clinical manifestations, whether it's muscle or CNS. And at the heart of what we are trying to do is correct that splicing to improve symptoms. Right now, as you know, unfortunately, there are no approved DM1 treatments that target the defect in the DMPK gene that are truly disease modifying. And just in general, one of the biggest limitations in therapies trying to solve muscle diseases has been trying to get those therapeutics into muscle in a robust and efficient and reliable manner. And so we really need treatments for DM1 that get into the nucleus of muscle cells, that reduce the toxic DMPK mRNA that resides in the nucleus, and then ultimately restore normal splicing and hopefully stop or even improve function. So our goal is to create that kind of therapy, and we have a very targeted delivery approach that I'll get into very briefly here. So we've developed something called the FORCE platform, which is the basis for the investigational medicines we're developing. There's 3 components: an antibody, a linker, and a payload. And as you can see here, this Y-shaped figure is a monoclonal antibody. And what we've done is taken the top portion of that monoclonal antibody, or an antigen binding fragment, an antibody fragment of Fab, and we've engineered it to bind very tightly to something called the transferrin receptor 1, or TFR1. And I'll talk a little bit more about that on the next slide. And we believe that by using this piece of the antibody, it has advantages over monoclonal. It's smaller in size, hopefully better muscle penetration. It doesn't have certain aspects to it that may help with tolerability and immunogenicity. We then have this linker for which we attach the antibody to the linker and then our therapeutic payload. And this is a clinically validated linker, which just means that it's used in products that are on the market right now, other antibody drug conjugates. And so this helps de-risk our overall platform. And then we are able to attach different kinds of payloads to the antibody via the linker. In the case of myotonic dystrophy type 1, we use an antisense oligonucleotide because ASOs are known to be able to get into the nucleus, and that's where we want our effect to be. For other diseases where you may want to attack things in the cytoplasm, we could attach an siRNA. And in other cases, we can attach small molecules. So this is a modular platform that's highly flexible, where the antibody and the linker remain the same, and we can attach different payloads. So the TFR1 is ubiquitous. It's highly expressed across muscle cells, including skeletal muscle, diaphragm, cardiac muscle, and smooth muscle. And it has a natural function in our biology, which is to maintain iron homeostasis. So we're really using the body's natural cell biology, taking advantage of this TFR1 receptor that's found on the surface of muscle cells. And we created our force conjugate to attach to that TFR receptor. It's brought into the cell and forms this endosome. And then the antisense oligo is cleaved within the endosome and travels to the nucleus so it can do its job. And it does this without destabilizing the membrane. And so we hope to see in the clinic a wide therapeutic index and durable target engagement. Quick update on the ACHIEVE trial. So we've been very fortunate that we've had amazing input into our trial design from key opinion leaders from around the world, many of whom are in this room today, and also from global advocacy leaders, as well as directly from patients and caregivers. And all of this advice has helped with the design of our study, the outcomes that we're looking at, and all the way down to the clinical trial protocol and even visit schedules, because we are trying to minimize what we know is always significant burden to patients and their families. At a high level, this is a snapshot of what our study is. It's a phase 1/2 clinical trial to evaluate our investigational medicine Dyne 101 in patients with myotonic dystrophy type 1. We're including patients who are ages 18 to less than 50 years of age, and we have about 72 participants that we plan to enroll in the study. The primary endpoint is safety and tolerability, but we're looking at a number of secondary endpoints, including splicing, changes in DMPK, and multiple assessments of muscle strength and function. This study is essentially in three stages. The first stage is a 24-week multiple ascending dose period, which has a placebo arm to it. The second is a 24-week open label extension in which all patients receive active drug after the 24-week placebo period. And then it's followed by a 96-week long-term extension. It's very important to us to collect data over a long period of time. And I'm very pleased to say that we will have initial safety, tolerability, and splicing data in the second half of this year. So later this year, we will have data to share with the community. If you want more information on the study, you can see at the bottom of the slide here, we have a clinicaltrials.gov identifier. You can go to the clinicaltrials.gov webpage to get more information and more details on our study. And also you can go to the Dyne website to get more information on our study. This is just a very, very brief snapshot of the clinical trial inclusion and exclusion criteria. I won't go into all of it here. Again, you can go to the ct.gov website to get the full list of criteria. And currently, this is a global study, and we have sites that are enrolling in Europe and New Zealand. So that's a snapshot, and that's a brief review of where we are with our ACHIEVE phase 1/2 clinical trial with Dyne 101. We've also continued to explore what our technology can do. And so this is data that has been recently presented, and I'm very happy to share this with you here today. So we've continued to see what the FORCE platform can do, and we've been exploring whether or not it has the potential to cross the blood-brain barrier. You've taught us very well the importance of being able to get into the CNS, being able to resolve some of the CNS manifestations of myotonic dystrophy. Not surprisingly, as you know, the brain is highly vascularized. There's a very, very extensive capillary network. And so this allows for a widespread delivery of therapeutic agents. And so the TFR1 receptor is found on the brain as well. And so we wanted to see if we could take advantage of this biology and see if the force conjugate can cross the blood-brain barrier. And as you know, the blood-brain barrier is there to protect the brain from molecules, large molecules that should not be entering the brain. However, there are times when you want therapeutics to enter the brain, and many other companies are also using the TFR receptor to actually target specific CNS disorders. This is an experiment in non-human primates or monkeys, where we wanted to understand the potential of our force conjugate versus an unconjugated antisense oligo, so we call it a naked antisense oligonucleotide, delivered intravenously, an unconjugated antisense oligo in the middle column, delivered intrathecally, so directly, and the force conjugate delivered intravenously. And what you can see here is with the naked ASO delivered IV, there's essentially no antisense oligo in the brain. When it's delivered via IT, you can see that there is some ASO delivered to the brain, but it's really at the superficial layer there at the top where you can see the ASO staining in the cortex region. And then with the force conjugate, we were really pleased to see robust ASO delivery into different regions of the brain, including the deep brain regions like the caudate and the putamen. We also wanted to see in another model, this is a mouse model, if there were any beneficial effects of being able to have this delivered to the CNS. So this is a mouse model that I've talked to you about before, I've presented. It's called the HTFR1 DMS-XL mouse. And really the big takeaway here is that this is a very, very important mouse model because it expresses the human transferrin receptor and expresses human toxic DMPK. So potentially there is very good translatability between what we see in this mouse model and what we may see in the clinic. And we ran a number of experiments. This is just one of those. This is data from the cerebellum. And I'll just kind of orient you to the slide here. The first slide is whether or not there's delivery to the cerebellum. Is it getting in the cerebellum? The sort of the middle part of the slide is, are we knocking down DMPK? And then the right-hand side, are we reducing the DMPK foci? And if you recall, you've heard about these trap proteins. Well, these trap proteins are called, develop foci in the nucleus of cells. And so the desire is to be able to reduce the foci. So what you can see here, if you maybe look at the red bar and the blue bar, the naked ASO versus force, we're getting much higher delivery of the ASO to the cerebellum. In the middle panel, if you look at the red and blue bars, there is significant decrease of DMPK in the cerebellum. And then we're able to reduce the foci by about 60%. Again, these are early data. We're just starting to explore the potential here, but we're really excited about this early data and the potential for CNS, for alleviating CNS manifestations. So overall, we believe the platform has the potential to overcome these barriers of oligonucleotide delivery to the brain and muscle. And we know we need to be able to treat the whole body to make a huge difference in myotonic dystrophy type 1. And again, with that, I just want to thank you for all of the sacrifices that you've made to participate in our study, for all that you've helped us learn about over the last several years. So thank you so much.
Good afternoon, everybody. My name is Aisling Holland, and it's my pleasure to be here today to give you a bit of an insight into the work that we'll be doing at PepGen and supporting the development of our compound PGN-EDODM1 for myotonic dystrophy type 1. But first, I just wanted to say thank you to the organization for inviting us today. I know that when we got the invitation, PepGen was delighted to be able to receive it and to be here today. But also thank you to the community and for all of their work and their input and their continuous conversations with us as we start to develop and build out our program as well. So I'll pause here. These are our disclaimers and forward-looking statements. So I think Andy gave a really nice introduction yesterday into the different types of therapeutics that are in development at the moment for myotonic dystrophy type 1. So there's different companies that are working at it at the DNA level, at the RNA level, or at the protein level. And for PepGen, we're looking at it at the RNA level. So really trying to address the root cause of the disease and really getting to the pathogenic side of DM1. Now, the way that we do this is using oligonucleotides or oligos. But unfortunately, one of the big challenges and the limitations for the development of oligos is the delivery capacity. Now, we believe that we've found a solution for this, and we've created our technology platform called Enhanced Delivery Oligonucleotides, or EDOs, that we can show that we have up to a 25-fold higher level of nuclear delivery into muscle cells. And what our EDO platform is comprised of is two primary parts. So the therapeutic part, which is an oligonucleotide that is primarily targeted to bind to the repeat expansion present in DM1. And the delivery capacity is really enhanced by our proprietary technology, which is our cell-penetrating peptide. And our peptide is a short synthetic peptide. So again, I know that I'm talking to a community that is all too familiar with DM1, and we've had lots of introductions to it from the molecular and the scientific capacity as well over the last few days. But just to put it into context and to provide a little bit more clarity into the approach that PepGen's using for tackling DM1. So DM1 is caused by a CUG repeat expansion, and that repeat expansion binds different proteins like MBNL. And when these proteins are bound, it results in abnormal splicing, which then means that different proteins are misprocessed and causes and contributes to many of the different symptoms present in DM1. However, with our PGN EDO DM1 approach, we have it designed so that it can bind to the repeat expansion and free MBNL1 so it can go back to its normal function. So when our EDO is bound, it binds to that repeat expansion, and then that can allow MBNL to be freed and restore the normal processing of proteins and restore the normal body process as well. So in order to evaluate this, over the last number of years, we've carried out a lot of different pieces of research to help provide a package of information to really show the support for the potential application and translation for EDO DM1. The first way that we looked at this is in human muscle cells. So we've taken sample of cells from an individual with DM1. And we've heard a lot over the last while that we can see in muscle cells, now we can see these foci or these repeats. And there on the left-hand side in the blue, it's those little red dots that we've tried to highlight with arrows just so that it's a little bit clearer on the screen. But also MBNL1 is also co-localized to these foci, and we can see that by those green dots. So when we do a single treatment with EDO DM1 in vitro, we can see that we can reduce the number of foci per nuclei. So we can address that initial point. And we can also liberate or free MBNL1. So it's no longer bound to that repeat expansion. And it's great that we can get that early mechanistic part, but most importantly, we're also able to see that with misspliced correction, when looking at a variety of different transcripts, we can also correct that missplicing profile from a global capacity. And again, for all of these evaluations, we can see a nice dose response, and we can see up to a 69% correction of missplicing in vitro. Now, in vitro is great, but we can see that the real challenge is when you go in vivo, so into animal models, and eventually when we get to the clinic as well, that delivery profile can be a little bit more hampered. So we've used the HSA mouse model, and I think Ami Mancodi and Charles Thornton are in the audience and have been here this weekend, and they generated this mouse model 23 years ago. It's a really well characterized and one of the most robustly used models in the field for DM1. And what's really nice about this model is that it retains a lot of the key phenotypes at the molecular level as to what we see in DM1. So we see the foci, we see the binding of MBNL1, and we can see similar missplicing transcript profiles. But importantly, we can see from a functional perspective if we can restore some elements of that. And this model can help us understand that from the myotonia standpoint. So our first evaluation, we looked at a single dose in HSA mice. And when we look at it 2 weeks post single systemic administration, we can see great correction in key muscle-specific transcripts. So when we look at two in particular, ATP2A1 and CLCN1, we can see that we get 91% correction for one of them and 68% correction in the other. So again, similar to what we saw on the in vitro side, we're able to translate that misspliced correction into the DM1 mouse model as well. But importantly, we can see that we can take it a step further and we can get correction of myotonia. So we evaluated this with the electrophysiology, so highly quantitative evaluation. And after a single dose, when we look at it 2 weeks later, we can see complete correction of the myotonic profile. But again, I think a picture paints 1,000 words and a video can be even more impactful. So on the video on the left-hand side, we can get it to play. Yep. We've got the no-treated HSA mice. So what we're doing here is we do a pinch test to see if we can see that myotonia. And we can see that quite clearly in the hind limbs. When we pinch it, they drag behind them. However, when we do a single treatment with EDO-DM1, on the video on the right-hand side, when we do the same pinch test, we can see that that myotonia, or that dragging of the hind limbs, is no longer present. So with our compound and with DM1, we know that it's going to be a chronic administration with the approach that we're using. So we wanted to look not just after a single dose, but after repeat administration, and to see, can we have an additive benefit with repeat administration? So with this, we looked at multiple missplicing profiles again, just like we did for the earlier work. And we can see that 4 weeks after a single dose versus 4 weeks after 4 administrations given every month, we can see that we've got an improved profile for missplicing transcripts. So we've got an additive benefit with repeat administration, bringing it from 68% correction from a single dose, looking at across multiple transcripts, bringing it to 99% correction with 4 doses every 4 weeks. And again, importantly, we wanted to see how does that look from the myotonia side. So this time we did it by pinch test, so like what we saw in the video as well. And we can see that after 1 dose, we've got, when we look at it 4 weeks post-dose, as opposed to 2 weeks in the previous slide, we can see 76% correction of myotonia. And when we look at 4 doses, we can see 99% of myotonia correction as well. And again, when we look at the tissue concentration, we can see with repeat administration, we're getting increased amounts in the tissue as well. And again, with the profiles that we're seeing, it's really encouraging for the potential translation side of things. And we heard yesterday that it's all about coupling the activity side of things with the safety. And because of the interest of time today, I won't be able to go into too much of that safety data, but I can say that it is all coming along and coming together very nicely, that it's enabled us to open our phase 1 clinical study in Canada. So we released this week that we had the good news that we've been awarded the opportunity to start our clinical trial, which is a phase 1 single ascending dose. So this clinical trial is called Freedom DM1. And we will be doing 3 different groups for cohorts for the single administration. So a 5 mg per kg, 10 mg per kg, and 20 mg per kg. So this study is going to be a randomized, double-blind, placebo-controlled trial. And we anticipate a variety of different readouts. So phase 1 studies are looking very much at the safety side of things. So that's going to be a big component of this study. But in addition, we're also going to be looking at other readouts, including the missplicing, so previous to what we've shown in the other slides as well, and also the functional assessments. So with this, we'll be doing different biopsies throughout the period of the study as well, so that we can really see the profile of that and how it translates into the clinic. Now, we're currently on clinical holds with the FDA, but we also got good news this week to say that we've been awarded orphan drug designation from the FDA as well for our PGN EDO-DM1. So I know this is open in Canada at the moment, and we're working closely to try get it opened in the US as well. So just as a quick recap of where we are from our program and from the development for the DM1 side of things. So as I say, our EDO-DM1 compound is really engineered to use a unique mechanism to liberate MBNL1 from the repeat expansion. And we can see in vitro that we're able to reduce the foci, liberate MBNL1, and correct the missplicing profiles. In addition, in HSA mice, for the DM1 mouse model, we can see robust missplicing correction, reversal of myotonia after single dose. And what I didn't show today is that we've got sustained correction out to 24 weeks. So that's incredibly promising if we can get that translation in the clinic as well, meaning less frequency in dosing. And again, with repeat dosing, we can see additive benefits and further enhancement of the correction of the myotonic profile. And as I say, we couldn't go into too much detail on the safety side of things, but what I can say is that it's well tolerated through 90 mg per kg for a single dose study in non-human primate studies, which is incredibly encouraging from the safety tolerability side of things. And so much so, as I say, we've been able to open our Freedom DM1 clinical trial, which will be open in Canada at the moment, and we anticipate the interim readout of data for 2024. So if we're invited back next year, hopefully we'll be able to share some insights into it then. So I will just pause there and say thank you very much to the community for having us here today. And thank you to the organizers. And I'll pause there.
Good afternoon, everyone. My name is David Seiden. I'm the Senior Medical Director for Clinical Development at Harmony Biosciences. I'm actually a neurologist, but I'm a sleep specialist. I'm not a neuromuscular specialist, which is sort of the reason I ended up at Harmony, as you'll hear in a minute. And I do want to tell you that I've heard some great data over the last few days, really interesting things, hearing this as a sleep specialist. And you may be a little inundated with data. I was told at lunch that you may be feeling that way. So good news is I don't have any data to show you, or I don't have any slides to show you either, but I do want to just tell you a little bit about Harmony and where we're at with our clinical trial, which the myotonic dystrophy community has been very supportive of. So at Harmony, we are a company that works to develop and deliver treatments for rare neurological disorders, sometimes disorders that are overlooked. And if you look at our website in terms of what we're working on, you'll see sort of a common theme that these are rare neurological disorders in which a key symptom of that disorder is excessive daytime sleepiness. That's how we come to the myotonic dystrophy community. But some of the other disorders that we've been looking at include Prader-Willi syndrome, another rare neurological disorder, idiopathic hypersomnia, and in fact, the medication that we are looking at is called pitolisant. This is a wake-promoting agent, and it has been FDA approved and is currently on the market, marketed as Wakix, and it's currently on the market for narcolepsy, of course, a disorder with significant excessive daytime sleepiness as a primary feature. We're very thankful to the Myotonic Dystrophy Foundation. Thank you. And the myotonic dystrophy community for their willingness to participate in our phase 2 proof of concept signal detection study of pitolisant versus placebo, in which we looked at both the safety and efficacy of that compound, specifically for excessive daytime sleepiness in patients with myotonic dystrophy type 1. Just a few words about pitolisant, Wakix, as it's marketed for narcolepsy. This is a wake-promoting drug. It acts by increasing levels of histamine in the brain. Histamine, as you may know, is one of the myriad wake-promoting neurochemicals in the brain. These chemicals also include something called orexin, and some of them and the monoamines as well. But pitolisant works through the histamine system. I'm happy to tell you that enrollment for the trial has finished. This phase 2 trial. We have a few participants that are still finishing up, and we are very hopeful that by the end of the year, we'll have some top-line data to present to you. I'm sorry I don't have any data to present to you yet, but we are just about there, so stay tuned. And again, we appreciate all your help in getting us to this milestone. And if anybody has any additional questions after this meeting, I'll be happy to talk to you. So thank you very much.
Hello, my name is Heather Sadlish. I'm an executive director at Arepta Therapeutics, and I'm really excited to be here today to talk to all of you. This is my first time at this conference, and I have to say that I really, really enjoyed it. Speak up? Okay. Sorry. Okay, sounds very loud to me. Thank you. Once I get used to the microphone. No, this is really exciting. I think this is a wonderful event. It's such a great opportunity for the researchers to meet with the patients and for the patients to meet with the researchers. I'm a scientist. I don't get out of the lab very often. And I've had a wonderful conversations with a number of patients, really understanding what they're going through and how they're surviving with these terrible diseases. So again, thank you so much to the organizers for inviting us. And I'm really looking forward to telling you our story. So we are targeting a specific part of the biology that we think is really fundamental to these diseases that have these repeat expansions, such as myotonic dystrophy. As you know from the various talks that you've seen over the last couple of days, myotonic dystrophy is caused by these repeat expansions that are found in the DMPK gene, these CTG repeats that keep expanding over the course of the patient's life. And these have an impact on the disease progression. Once these repeats have been forming and creating these large expansions, they alter the RNA structure. And this altered RNA structure then leads to sequestration of various splicing factors that are really important. And with that sequestration, it has this dysregulation on the splicing events. And so this is where you have these many, many different types of clinical characteristics that are the result of these changes in the splicing events. So over the last couple of days in the industry updates, you've heard from many companies that are targeting many different steps of this process. They're knocking down the DMP expression, they're modulating the sequestration of the splicing factors, or they're modulating downstream effects such as the sleepiness. So what we think differentiates Arepta's approach is we're going at the very beginning. So we are targeting the somatic expansion. The expansion is repeats over time in the patient. And we think that that will have an impact and enable slowing of the disease progression. So how does this work? So while myotonic dystrophy and DM2 are perhaps less well studied than we'd like, there are lots of other diseases out there that share the same characteristic of having these repeat expansions. And from these other diseases, we can learn a lot. So one of these is Huntington's disease. And there's been a lot of active research on Huntington's patients and using sequencing of these Huntington's patients to understand what genetic variations lead to changes in the onset of that disease. So some alterations lead to a later onset or an earlier onset. And from this information, you can hone in on certain parts of biology that we think are really important for the disease. And so there was a really pivotal paper that was published a number of years ago that identified a series of proteins called, they're involved in DNA repair. And so this is shown on the graph on the right. All these different proteins form these complexes. And these complexes have a fundamental role of repairing the DNA. And the problem with the repeat expansions is the DNA is a bit slippery. And so that repair process is somehow flawed. And so we think that based on these studies, as well as other studies, that by targeting the DNA repair process itself, we can actually stop the repeat expansion. So one of these proteins in the DNA repair process is called PMS1. And so PMS1 is the one that Arepta is going after to say, if we can stop that protein, then we can stop the somatic expansion. How do we do this? So we are focused on our own splicing modulation. So this is different from what you see in myotonic dystrophy. From splicing, from your gene, so from the PMS1 gene, you have the RNA that's being generated. And that RNA is spliced to normal RNA. That then becomes protein. That protein then is involved in the mismatch repair. And that mismatch repair is driving the somatic expansion. In the RNA for PMS1, there are these weak exons. So this is a sequence that's not normally present, not normally incorporated into the final mRNA product. And what Arepta has done is identified ways to increase this incorporation. So our small molecules, our compounds, bind the splicing machinery and alter it just slightly. And so what that leads to is mRNA that has that weak exon or that sequence that's not normally there. And that alters the whole process. And so what you end up with is an unstable protein. So it's not present. So the impact of that is that the mismatch repair doesn't happen and you don't have somatic expansion. So this is a kind of a novel approach. And we have been working hard on this. So this is a very, very simplistic structure of what's involved in the discovery of a drug. And I think my talk is very unique from the other ones here in that we are at a very, very early stage. So you need to have those compounds that you screen. And when you've screened them and you find ones you think are interesting, you have to show that they work first in Petri dishes and cells, then you move to animal models. Then eventually you can build up enough data that you can take things into humans. So we are still very much in the early stage. We are making progress, very much so. But what I want to show you is some really exciting data where we have taken a look at our compounds in cells. So these are cells that have been collected from a DM1 patient, and that patient has about 238 repeats. And these cells are embryonic stem cells. So they aren't a neuron, they aren't an astrocyte, but they're a cell that we think is reflective of the disease, because what we can do is measure the expansion over time. So what we can do is actually collect the DNA and over time see what happens. And so over a period of about a month, we see that the 238 repeats expands by 13. And so this is significant enough for us to be able to measure pretty accurately, and we're pretty confident of it. And so when we set up this experiment with our compound, we wanted to see if it stopped that expansion. And our goal was, can we reduce it by half? Because if we can reduce that expansion by half, then we feel pretty good about it. And in fact, when we treated these cells with our compound for a month, we only got 2 repeats. So we really, really, really had a big impact. So we're pretty excited about this, because if we can then show that this works in animals, and then we can get something that's safe enough, we can try it in patients. It's still an untested theory. There's a lot of evidence from the literature that by altering these DNA repair proteins, we can have an impact on somatic expansion, but it still needs to be proven. So at the end of the day, Arepta is taking a very different approach from the other companies. We are targeting that root cause, the actual expansion itself. There's a lot of data out there from different diseases that are very similar with the repeat expansion part that suggests this should be feasible. We are the first to actually have molecules that can do this, that can stop the PM1 protein being expressed, PMS protein from being expressed. And we have data in cells that shows that we are reducing the repeat expansion as you would expect. So what we are hoping that we can provide to you at some point, once we have all the safety data and important studies that are necessary, is a small molecule as a compound that you can take orally. It will get into the brain and it will reduce that somatic expansion. And we hope that that is going to slow or stop the progression of the disease. So thank you very much for listening. We are a small company and really excited to be able to contribute to this process.
Well, good afternoon, ladies and gentlemen. I hope we're all feeling very rested after lunch. My name is Ibs Mahmood, and I'm the CEO at Amo Pharma. And look, before I even get started with what I'm going to talk about today, I do want to thank Tonya and the whole team for laying on this wonderful event. So thank you very much. And that's not the last clapping you're going to be doing today. So look, I'm here. Do you know, I used to stand up a lot and talk in conferences in my last job. I can't remember the last time I stood up feeling this excited, because I'm about to tell you about phase 3 trial results that we've just had. And well, I'm really looking forward to showing it to you. But look, if this works, Yeah. Before we do that, there are some thank yous I need to do beyond the MDF. So look, you can't run a phase 3 trial without the investigators, without those wonderful doctors that you've been meeting throughout this event. They were remarkable. So I do want to thank all of them and all of their teams, the enormous amount of admin that goes into a clinical trial. I want to really say thank you to our vendors and partners, the likes of Styrus, who I know are here today, who did an exemplary job. I do have to thank the money guys. It does cost money. So we had some wonderful investors. But the really big one I want to thank are the people who took part in the study. And you know what? There's a preface to that. There's a backstory before I even get going. I don't know how many of you were in this room yesterday and at the FDA panel, and there was a veterinarian called Mark, and he stood up and he talked about the progression of his disease. And he talked about how he was worried for his kids, right? Now, I'm a dad. I have 5 beautiful kids in Ireland, and they're healthy. And it really moved me to think he's thinking about them. This is a man who could run these incredible marathons, and he wasn't thinking about himself at all. And I'm about the same. I'm sad to say I'm about the same vintage as Mark. And so that really struck me. But you know what really struck me was that the nature of this condition is absolutely exhausting. It's like he'd run a marathon even before he started talking. And despite all of that, he then stood up, he made his presentation, and then he challenged, this very powerful government agency to try and give him a drug, right? So I was blown away. I can't even pretend to imagine what it would be like if one of my kids had it, let alone to run a marathon and then to say something as strong as that. And then he was followed by the 2 ladies who, I'm sad to say, I think I could be old enough to be their dad. And they made the same story. They told these incredible anecdotes about their lives. And then they went on and said, look, please give us the chance for medication. Look, the point of telling you all this is that I think it's incredibly brave to do this once you've been through this condition. So I really want to thank the entire patient population for fighting, for, despite being so tired, trying to do something about this condition. And it galvanizes me, I have to say. It galvanizes me to make sure that we do something about this condition, right? And I would say, I need to tell you that almost everyone that works at Amo Pharma has a child with, I call them special, so a neurodevelopmental condition. They don't have DM1, but something similar. So we really get it. So I think that the people who went even further and participated in a phase 3 clinical trial are heroes. So I want to thank you all for that. So sorry for the long anecdote, but thank you. Right. So let's talk about the bit you really want to hear about, which is the REACH CDM study, which I said was a phase 3 study. And here's the punchline. It showed benefit across the body of the bodies of these patients. So we're not just talking muscle effect. We're talking a change in the IQ of these children. We saw real world changes in their social abilities. We saw changes in their blood markers. We saw children who'd never spoken before talking to their parents. It was an utter privilege to be involved. And the next step is we're going to take this into adults. Okay. Tanya's sitting there thinking he's never going to get through this in 10 minutes. Don't worry, I'm going to speed up. This is one I can go fast on. You all know what myotonic dystrophy is, but I do want to remind you what congenital myotonic dystrophy is, and in particular, why we decided to study it first. With a lot of people here having adult, and it's simple. It's back to my introduction. The kids are sicker. So we wanted to go after them first. And because they're sicker, we believe that if we can help them, it's really likely that we can help you. So that's why we did it that way around. There are some technical reasons as well, like it tends to be more similar in the way that it presents, so it's easier to run a clinical trial. The way that we define congenital myotonic dystrophy is that you have to show the symptoms within a month of birth. And you have to be diagnosed by the time you're in the clinical trial. So that is the area we're going after. And as Andy presented so clearly, that it's caused by the presence of this nasty abnormal RNA. And we'll talk more about that in a second. So again, there's been a lot of talk about the science today. I need to beat Andy up slightly because when he presented us earlier, he said that we didn't interact with the RNA. In fact, that's the main thing we do. So our very simple molecule, very small molecule, gets into every cell in your body, as far as we know, and locks on and destroys this junk RNA. And what this whole slide basically says is it disappears. So you see the red coloring, that's work by Professor Wang, who's probably in the room here. He stained this nasty RNA and he showed that after the addition of the drug, whether or not it's in human tissue or in animal models, it's wiped out. So that's how the drug works. So we ran a phase 2A study. So that's just a small study to see if this thing potentially is going to work. And what we saw back then in 2017 was that there was effect right across the symptoms of these people in this population in the UK. This presentation, I believe, will be online afterwards so people can look at the details, but that's the bottom line. You probably want to hear about the phase 3 more than this. So this is the phase 3. So it was a global study. It was called REACH CDM. It was for the congenital population. It was girls and boys between 6 and 16 years old, and they had to have a certain level of the condition to be in the study. It was a fairly typical design. So you have a group who get the drug and a group who get the sugar pill, the placebo. And then we wanted to see if there would be differences in these various measures. Now, the primary thing, and when you go into a phase 3 study, you have to tell the Food and Drug Administration what it is that you want to be judged on. And that is called the primary. And we used a rating instrument. That is like a checklist. Psychiatrists use it all the time. And it's a long list of things. And the reason we did that is we wanted to capture the whole of the condition because it's so varied, right? Some kids have trouble with their muscles. Some kids have trouble with the way that their brain functions. It's so variable. So we wanted to make sure we captured the whole condition. So we used this very novel rating scale in this particular clinical trial. But thankfully, we also measured a whole bunch of other stuff. And I would describe these as being very computer-like. They are just simple tests that measure something in a completely objective way. So there is no sense that the physician is involved in subjectively measuring things like in the rating scale. So these are things like how fast can they walk 10 meters or how strong is their grip? And we saw those videos so clearly yesterday. So there's a whole bunch of those things as well. This is a table that just shows who was in the study. I'm not going to go through it now, but all it really says is it was a fairly balanced study. It was fairly representative of the kinds of people that have the condition. And these are the results. So the first thing I want to tell you is a little bit of bad news, which is, remember I talked about that rating scale? Well, what we saw on that rating scale was a significant placebo effect. So both the group who got the drug and who didn't get the drug got a lot better according to that scale. And we were scratching our heads as to why that could have happened. And I think we're now fairly sure that what happened was that we were being too ambitious. It was a complicated scale being given to a patient population that never had it before and a group of doctors that never used it before. And on top of that, this thing we never talk about anymore, COVID. So we weren't able to sit next to these physicians and make sure that it was being implemented properly. So it wasn't. And I have to hold my hand up for that. So then we looked at all the other things. You remember these things which are nothing to do with an opinion of anybody. They are just measured. And 10 of the 12 things that we measured worked. And I am so proud and excited to say that. And again, it's all over the condition, right? So there's 4 different things that look at muscle function: the 10-meter walk test, the grip strength, the lip strength, right? A scan of your muscle strength, and even if you take the blood of these individuals and you measure a particular protein to do with the breakdown of muscle, that improved as well. But it's not just muscle. As I said, this drug gets everywhere. So, some of the earlier presentations, they're doing great work on getting it into the brain. This drug is in the brain, right? So we saw remarkable effects on how the brains function in these individuals. And then we saw the next level, sort of beyond brain function, which is the social aspects. The only thing we didn't really see any change on was bone density. Our guess is that the 5 months of the study wasn't long enough to see bones getting stronger, but we're not sure why. So could that result just have been 1 or 2 patients that got better? Could that result have been that there was just 1 or 2 facets of the condition that got better? Well, this pretty set of tables hopefully answers that question. So along the side are different patients, and along the top are very different measures of their condition, right? So we've got 10-meter walk, grip strength, IQ, etc. The left-hand table is the people who didn't get the drug, and the right-hand table are the folks that did. And the ones in purple got better. So what you can see immediately is there's lots more purple on the right, which is great. It means that the people who got the drug got more better than the people who didn't. But you can also see that the purple isn't concentrated anywhere. It's not one patient that got better. It's a variety of patients. And it also isn't in one column. So it's not that just people got better on the 10-meter walk test. Some of them, their IQs went up. Some of them, their 10-meter walk test went up. Some of them had both. You see, this is the challenging thing about this condition. It's so varied. You need to do quite tricky things to make sure you capture it all. But I just love this diagram because it's blindingly obvious. It works across the piece. So if you do an analysis that's beyond my ability, but where you essentially bring all that data in that previous table together into like one whole analysis, and you let the mathematicians loose on that, then they will tell you that there is no way that it's a fluke. So we talk about whether something is statistically significant or whether it was a fluke. And the asterisks on this diagram simply say it wasn't a fluke if you bring all the data together. So that is a huge relief for us because it gives us a strong argument with the Food and Drug Administration. And I guess these are the measures which we think are absolutely critical to the next stages of this condition, in particular when we move into the adult population. So what about safety? The first thing I would say is this drug has been in between 500 and 600 people, and to date has had no significant health safety issues. And that was repeated in this clinical trial. There was nothing that came up that worried our chief medical officer or indeed the regulators who regularly checked in with us to say it was safe. There's an interesting one, though. If you look really carefully, you'll see that if you look at diarrhea there, the placebo group is way higher than the active group, right? And diarrhea being a side effect of potentially of the drugs. So what's the explanation for that? Well, gastro issues are common in DM1, right? So they just got better. That's why there's less diarrhea in the active group. Yeah. So it's a weird safety signal hidden away it's a weird efficacy signal hidden away in the safety data. So what are we going to do next? Well, we need to complete our analysis. We need to design an adult study, and we need to contact the regulators and sit down with them and work out what we're going to do next. If I may just be a bit blunt, I suppose, it works and we're really excited. And, you know, as I said, everyone in the company has a kid with a similar sort of condition. So this was a labor of love. And so 8 years of ups and downs, and I'm really excited to say it works. But we live in a highly regulated world. And so I don't want to misrepresent that we do need to work with FDA to see if they will, potentially conditionally approve this drug based on the fact that there's clearly something very exciting going on here. And I'd like you all to just bear with us as we go through that process. But, you know, I think if there was a moment for I think we wouldn't exist if it wasn't for you. And, you know, perhaps this final step, there's a moment for the patient groups to really think about the chances that this thing gets approved. Even if FDA doesn't let us go through, then we will still be running the adult study in it either way, right? So the adult study will either be after an approval or before an approval. But either way, we have to, after Mark and Haley and Janine's, testimony yesterday, we owe it to all of ourselves to run that adult study so that we will have a single, simple molecule. It's just a strawberry flavored drink. It's really easy to take, that goes a long way to fixing this condition. If you have any questions, please come to the store, which is right outside the door. You may want to know about the adult study. And we're also hosting a breakfast tomorrow morning. Thank you very much.